All right, everyone, welcome. Today, we're diving deep into the most fundamental unit of life, the mammalian cell. And look, I know this can feel like basic science, but trust me, mastering this stuff is everything. It's the unshakable foundation you're going to build your entire clinical career on. So let's get our tour started. Just think about that for a second. A nerve firing, a muscle contracting, your immune system fighting off an infection, how a patient's body metabolizes a drug you prescribe. Every single one of those processes, whether it's normal physiology or a disease state, starts right here at the cellular level. We're about to explore a tiny, living city just bustling with activity. And if you can understand its layout, you can understand the whole human body. So, first things first, every city is built out of something, right? Our first stop is to check out the basic building materials of our little cellular metropolis. When you put all these substances together, we call it protoplasm. Now, one look at this, and you see the cell is basically a watery world. I mean, up to 85% of it is just water. But don't let that fool you. The real magic is in that other 15 or 20%, the stuff that's floating around in that water. That's what gives the cell its structure, its function, its life. It's the soup where all the chemistry happens. This chart really makes the hierarchy crystal clear. After water, proteins are the undisputed VIPs of the cell, making up a huge 10 to 20% of the mass. And that makes perfect sense, because proteins are doing two massive jobs. They're the structural stuff, kind of like the steel beams and girders holding the city together, and they're also the functional machinery, the enzymes that make almost every single chemical reaction in the cell actually happen. Okay, let's move to our first major landmark in this city tour. We're going to look at the cell's outer boundary and the internal framework that keeps it from just being a shapeless blob. Think of these as the city walls and the internal support beams. And here it is, the cell membrane. It is incredibly thin. We're talking 7 to 10 nanometers, but don't underestimate it. It's a powerful barrier. Its core is this lipid bilayer, basically a two-layered film of fatty molecules. And that fatty nature? That's the whole point. It naturally repels water-soluble things like ions and glucose, which gives the cell total control over who gets in and who stays out. So, if the lipid bilayer is the wall, how does anything useful get through? Well, that's where these proteins come in. The integral proteins are the big players. They punch all the way through the membrane, acting like tunnels, gates, and selective carriers for specific molecules. They're also the cell's antennas, acting as receptors for signals. The peripheral proteins, on the other hand, just kind of hang out on one surface, often acting as enzymes or helping to control what the integral proteins are doing. Now, inside the city walls, you've got the cytoskeleton. And listen, this is not some static scaffold. It's a totally dynamic network. You've got these flexible actin microfilaments giving the membrane support, super strong intermediate filaments that are like ropes, giving the cell strength against physical stress, and then these rigid microtubules. Think of the microtubules as a highway system or a conveyor belt that's constantly shuttling organelles and other cargo all around the cell. All right, let's head deeper into the city, down to the factory floor. This is the industrial heart of the cell, where all the manufacturing, processing, and shipping of its most critical products takes place. Meet the endoplasmic reticulum, or the ER. It's this huge network of interconnected membranes. The difference between the two types is simple, but it's absolutely critical. The rough ER looks rough because it's covered in these little dots called ribosomes. This is your protein factory, the main assembly line. The smooth ER, no ribosomes. This is where lipids are made, and here's a key clinical point for you. It's where a lot of drugs and toxins are detoxified. You know, the logistics of getting a protein out of the cell are just incredible. It starts its life on a ribosome, gets folded up inside the rough ER, and then it buds off in a little bubble called a transport vesicle. That vesicle then travels to the Golgi apparatus, which is basically the cell's post office, where the protein gets processed some more, sorted, and packaged into a final secretory vesicle, all ready to be shipped out. It's a beautiful, seamless workflow. Now, no city can run without essential utilities, right? You need power and you need waste management. So let's go visit the cell's utility plants. Just let this number sink in for a second. More than 95%. Over 95% of all the energy your cells get from the food you eat, the energy that powers literally everything they do, is generated by just one type of organelle. And here they are, the famous mitochondria. These are the powerhouses of the cell. They take fuel, like glucose and fatty acids, and through a series of chemical reactions, they churn out ATP. ATP is the universal energy currency of the cell. It's like cash for getting work done. And it's why cells that have huge energy demands, like your heart muscle cells, are absolutely jam-packed with thousands of them. And here's a wild fact. They have their own DNA and can make more of themselves whenever the cell needs more power. So what about keeping the city clean and safe? The cell has two specialized cleanup crews for that. First, you've got lysosomes. Think of these as the city's recycling and waste disposal centers. They're filled with digestive enzymes that break down old organelles, bacteria, whatever junk needs to be cleared out. Then you have peroxisomes. These are the hazardous waste specialists. They contain enzymes called oxidases that neutralize poisons. And here's your classic clinical pearl. About half of the alcohol a person drinks is detoxified right here, inside the peroxisomes of their liver cells. Okay, we've arrived at our final, and you could argue, most critical stop on our tour. We're heading into City Hall, the central command center where every major decision is handed down. We're talking about the nucleus. Let's be clear. The nucleus is the absolute boss. It doesn't just store the blueprints, it actively runs the whole show. Inside, you'll find the DNA, the master genetic code. These genes dictate the identity of every single protein the cell will ever make. And by doing that, they control everything, its shape, its job, and when and if it reproduces. So what's inside City Hall? Well, it's protected by a nuclear envelope, a double-walled fortress with thousands of nuclear pores that act like security checkpoints. The DNA itself is carefully packaged with proteins into a complex called chromatin. And then you have the nucleolus. It's not really an organelle, but more like a factory inside the command center that builds ribosomes, the very protein-making machines that will then go out into the city. How cool is that? The command center builds its own factory workers. When it's all said and done, this quote says it best. The nucleus has the ultimate authority. It's the command center that sends out every single order that governs the cell's entire life cycle. Grow, mature, divide, or even, when the time comes, die. And this brings us right to the heart of why this matters for you as a future clinician. We've just toured this amazingly complex, perfectly balanced city. So what do you think happens when there's a blackout at the mitochondrial power plants? Or the lysosome recycling system breaks down and trash piles up? Or what if there's just one little typo in the master blueprint, the DNA inside the nucleus? The answer, of course, is disease. Every single disease you will ever study, from a simple genetic disorder to complex cancers, is, at its core, a story of a cell not working correctly. That's why we took this tour, because truly understanding the healthy cell in all of its incredible detail isn't just about passing a test. It is the absolute non-negotiable first step to being able to diagnose and one day treat the diseased one.